right, look, thank you very much for the opportunity to speak this afternoon. When I was given the topic for my presentation, Genomics England, looking to the future, I wasn't sure whether you were actually interested more in just what the view of the future is in terms of what Genomics England's going to do next, or whether actually I should be coming to a pharmacogenomics conference and talking about what the view to the future of that is. I've been caught with that latter approach once before, where 20 years ago I was invited to give a talk about male osteoporosis at a conference in Spain, prepared a presentation entirely about male osteoporosis to turn up to the conference to find out that the entire conference was about male osteoporosis, which was a bit embarrassing. All right, so caveat number one is I'm not a pharmacogeneticist. I'm a rheumatologist, and of course rheumatology is a profession and well known for poisoning their patients with things that are genetically influenced. Actually, more so, I'm actually a geneticist. What I'm going to present today about is I'm going to talk about research that has been done, particularly under Mark Caulfield's aegis as the CSO at Genomics England, and try and draw some lessons from that for you about how we might think about how best to go forward with pharmacogenomic screening. And then also present a bit about new programs that Genomics England's got coming forward, which I think will inform quite a bit of the what needs to be done in terms of development of service delivery and improvement of pharmacogenomic diagnosis. All right, so first up, for those of you who are not so familiar with it, the Genomics England main contribution so far to I guess genetics research overall has been the completion of the 100,000 Genomes project. So Doug, you asked for a data set that had genotype plus tumour sequencing. Well, this is the world's largest whole genome sequencing tumour normal cohort, and its second biggest number of cancer is breast cancer. So that's where I suggest you go. All right, so it studied from 2013 forward through to 2019, 100,000 roughly or so individuals. In fact, it's a bit of a misnomer, it's 120,000 genomes, and it's slightly less than 100,000 individuals, most of whom had rare diseases and some of whom had cancers. And so there are now, or rather to this point, there were 72,000 rare disease participants across a whole variety of different rare diseases. These are predominantly parent case trios, but there are a range of different family structures, and there are both childhood and adult onset rare diseases in amongst those. And then on the cancer front, we've got huge numbers of cancers. They're all tumour normal sequenced off fresh frozen samples for tumour at 60 times whole genome and 30 times whole genome for the normal. And these have all been reported back on through the NHS. This is now a living data set in that we are now ingesting additionally NHS Genomic Medical Service sequencing into this, and the very first bunch of those sequences have just entered the National Genomics Research Library today. This data set is publicly available. Of course, there are governance restrictions and checks about getting access to it, but anybody in the audience can access it. If you're academic, no matter where you are in the world, the charge is zero. If you're a commercial company, there's cost recovery involved in it, but it is not substantial. So this is a massive global resource. All right, so moving on then to pharmacogenomics. As I mentioned, Mark drove this program which looked, I think, starting around 2019, 2020 at the prevalence of pharmacogenomic variation within the 100,000 genomes cohort. So this particularly looked at unrelated cases, be whether they had rare diseases or cancers, and it filtered down through both an evidence-based set of genes and variants, and through a publication search and through expert evidence, expert advice, as well as looking at the CPIC guidelines to come up with a total of 63 gene-drug pairs, which actually includes all CPIC guidelines currently except for, I think it's CYP3A4 and a couple of HLA alleles. So it's actually pretty current for CPIC guideline pharmacogenomic variation. So they were all sequenced, and then we looked at the cumulative plots for, the frequency distribution plots for numbers of variants per individuals, and you can see that basically on average, the median number of pharmacogenomic variants per individual was 4, and almost 100% of participants had at least one pharmacogenomic variant. So on average, the 4 pharmacogenomic variants that were identified involved 3 genes and 17 different drugs, indicating that if you do whole genome sequencing, you pick up an awful lot that's relevant to almost your entire population. Now, of course, quite a few of those drugs are actually used in only a very small subset of the population, so a lot more needs to be done to justify doing this on a population scale. But I'll show you in subsequent slides why I think sequencing is going to be the right approach here and not SNP microarrays. Okay, so we also looked importantly at the different ancestral diversity of our patients, and this slide here, the blue, the dark blue are the African ancestry, African continental ancestry patients. The green are the Europeans, the yellow are the South Asians. We have some East Asians, but not a lot. And what you can see is that there seems to be an overrepresentation of African patients amongst those with rare variants. That is the case, but when we look, if you divide it up specifically, so you look at the prevalence of variants according to continental ancestry, the Africans do have more rare variants compared with, for example, the Europeans, but it's not that striking. In fact, one of the surprises of this is that although they have roughly 20% more total pharmacogenomic variants, it's not massively different in total number. However, when you look at the distribution between the different ancestral groups, they're wildly different. And that suggests that these pharmacogenomic variants are under some external evolutionary pressure. All right, so I'm going to show you some incredibly busy slides, and I apologise that it's mid-afternoon, a warm day. You're probably all looking forward to a bit of a snooze, but I'll try and pull some key points out of this. So in this figure, we've got in the top, basically these are all different genetic variants, and we've got the minor allele frequencies listed in numbers, and the height of this represents the minor allele frequency, and the genes involved across the top here. So the key thing is that if you look at the vast majority of these variants, their frequency is less than 1%. So the take-home message here is by far the most common group in terms of total numbers of variants in the community are rare variants. So it's not like common diseases where the majority of common disease risk is actually determined by common variants, pharmacogenomic variants, it's predominantly rare variants. Of course, there are some high prevalence common variants that are important, like the CYP3A5 variant, of course relevant to tacrolimus, the warfarin-related variants and things like that, which I'll talk a little, and the IL-28B, which I'll talk a little bit more about in subsequent slides. So the other thing to point out is that if you look at the colours across these, the colours vary significantly between them, and so interpretation of these in terms of their significance in finding them, like if you find a rare variant that is truly rare in your population, then it's much more significant than it's actually a common variant in your population. This is important for interpretation of these, but perhaps more important is the discovery, the fact that we're going to have to do a lot more discovery research, because the sample sizes for pharmacogenomic studies are actually still very small, and trying to identify things that are less than 1% prevalence robustly actually takes very large sample sizes. And I would argue that for most drugs, that's actually just not available currently. All right, so these are the variant frequency plots for the allele frequency of those variants in the genes that I just showed you, and the point here is that the vast majority of them are rare variants with a frequency of less than 1%, and as I'll show you, those sorts of things are extremely difficult to genotype by SNP microarray. All right, now here's a really busy slide for those of you who definitely needed putting to sleep. But there are only a couple of things that I want to show you on this. This is the previous slide broken up by different ancestral group and by different genes and different variants. And I'm going to highlight, first of all, the IL-28B variant, which, blowing it up a little bit. So IL-28B, or interferon lambda 3, is of course of relevance to ribavirin and PEG interferon therapy for hepatitis C. And the point about putting this one up is that this group here, which are East Asians, have extremely low prevalence, whereas Africans have extremely high prevalence of this variant, which results in lower response to treatment. And this shows basically that you need to, that although if you genotype this, it would, if the person had the relevant variant, even if they were East Asian or African, it would still basically indicate that they were less likely to respond to ribavirin. This actually, or peginterferon, this one variant actually is responsible for most of the ethnic variation in response to those treatments. So here we've got a really interesting drug-gene pair where the pharmacogenomics is almost monogenic. On the other hand, CYP3A5, the tacrolimus one, that's quite different. And here, most services that do pharmacogenomics type for the CYP3A5 number 3 allele, which results in basically no expression of CYP3A5. And so that results in a need to change tacrolimus dosage. That's actually less common in people from Africa, and it's thought that that's the case because there's, that gene is actually involved in protection against dehydration through an effect on salt metabolism, and that's resulted in maintenance of being able to express C3A5 being preserved in Africans. And what you can see is that that then drives a couple of other functional variants being relevant. So if you genotyped an African transplant population, for example, before you were about to give them a transplant, like, for example, one of the hospitals I work at, King's College Hospital, does a lot of renal transplant in people of African descent in South London. If you just genotyped this particular variant, then you'd be missing almost the same number of people who were requiring tacrolimus dosage because you'd only used a white European defined pharmacogenomic test. So Ancestry, particularly important. G6PD, this one's really quite interesting because if you look at all of the variants across G6PD, all of the ones that we found were very rare except for this common variant found in people of African descent. But actually there's another variant also that turns out to be highly prevalent in Africans. So if you just said, okay, well, we're only going to genotype the ones which have got prevalent in more than 1% of the samples, then the problem is that you wouldn't be genotyping this one. And this is another busy slide, but that one is found in roughly 24% of people from African descent. So again, it's just another example of why when you do this, you absolutely have to take into account the ancestral background, particularly in your discovery program. However, if you look at East Asians, they have a bunch of rare variants which are all completely different to the West Africans. They don't share, sorry, to the Africans rather, so they don't share any variants with the Africans, indicating that if you just studied Africans for your discovery set, basically you'd be missing a whole bunch of people from other populations who you should actually still be genotyping. All right, so HLA typing so I'm actually predominantly an immunogeneticist, and I've spent a lot of time using SNP microarrays for doing HLA imputation. And you'd be aware that Illumina, amongst others, have produced commercial arrays which cover a wide range of SNPs that are found in the CPIC and other guidelines, and also have dense MHC coverage to enable HLA imputation. The problem with this approach is that HLA imputation isn't that accurate. And so in our research programs, HLA-B and HLA-DRB1 imputation accuracies are typically in the region of 96 to 98%, which is okay for a research study, but for clinical implementation, I'd say it's probably not. And the other problem is that the reference panels for doing the imputation are almost entirely derived from people of European descent. And when you apply those to people of East Asian descent or African descent, the imputation accuracy drops significantly so that you're down more to around, for Africans, around 90%, and for East Asians, around 92 to 95%. And that level of inaccuracy, I would say, is almost unacceptable for research, but certainly would be for clinical purposes. And the variants are actually remarkably common and involve a whole bunch of different genes that are drugs which are of quite common usage, including anti-epileptics, antimicrobials, and then the other one I was going to mention was CYP2D6, which for those of you who are true pharmacogeneticists, this is really home territory for you, and you'd be aware that, of course, there are alleles of this that are actually virtually impossible to genotype by SNP microarray because they're long deletions. And so basically these can only be picked up by sequencing, and they affect a whole range of different drugs. There's another slide showing others that are basically HLA alleles, clozapine, allopurinol, and I'm really hoping that that slide will progress on. All right, so why can we not genotype these by SNP microarray? Well, firstly, if they're unreported so far, the only way you'll pick them up on a SNP microarray is to impute them. And if they're unreported and rare, it's highly unlikely that you'll be able to impute them with accuracy. Even if you directly genotype them on a SNP microarray, if it's a rare SNP, there's a high chance that you'll get your genotyping wrong. So this is a typical SNP microarray. And that's my finger there just to show you the size of it. As you know, basically each dot represents one SNP. One allele would be called green, the other one red, where they're both, where the person's a heterozygote, they're called yellow. And so therefore you end up with these cluster plots, which basically plot a red axis versus a green axis and cluster homozygotes, and in this case, for what I actually know is a rare variant where we only had 2 of the heterozygotes. These are heterozygotes. But how would you know that this is actually, these are rare variant single carriers, or that these are actually heterozygotes? And this problem actually often occurs with SNP microarray genotyping. Because you have so few subjects or that you're genotyping that fit into each of the 3 clouds, the rare homozygote, the heterozygotes, and the common homozygotes, it becomes very difficult to know whether your clustering is actually accurate and whether, in this circumstance, you're actually looking at 3 different genotypes or 2 genotypes. In fact, I know from sequencing that these are 2 genotypes, that these are heterozygotes, but you'd be hard-pressed looking at the clustering to know that. And that's going to occur because the vast majority of your variants are rare variants. That's going to occur a lot. And so you can get around this by running genotype known controls. But the problem with that is you would then have to run genotype known controls that cover all of your rare variants, of which there are far too many, so you can't actually possibly do that. So I don't see that genotyping is actually going to be the way forward for this. Instead, it has to be sequencing, and so thankfully sequencing costs are plummeting. They plateaued a lot over the last few years, we suspect because of lack of competition in the sequencing industry, in that we have essentially one monopoly player. That is changing currently, and we have both new competitors in short and long read sequencing, which I think is going to result in a substantial drop in sequencing costs, with a recent company launching, suggesting that they're going to be able to launch a commercial product in the next couple of years at $100 per sample, which is actually getting down into the cost of your standard pathology testing. So, for example, I know that the disease that I mostly work on, ankylosing spondylitis, a pathology lab test in Australia for an HLA-B27 is $160, whereas shortly we're going to have a whole genome sequence for $100. So I think at that point, basically, it's going to wipe out the array market for doing this. Right. Okay, so what happens next? What's GEL doing next, and how's that going to help here? So we have 3 new initiative programs that have been funded this year and will range out to finish in 2025. They are a genetic diversity study, a new cancer study looking at long-read sequencing technologies and a different model of doing cancer sequencing with sequences in local hospitals, and thirdly, a newborns program. And all of those, I think, have some relevance to pharmacogenomics. The diversity study, which I've put up here, I don't need to go through all of the work streams on it, but the main thing is that it's going to sequence probably 25,000 South Asian and African ancestry Britons, and we are hoping going to have a specific stream of pharmacogenomics working with the MHRA, we hope, to do major adverse drug reaction sequencing, particularly focusing on ancestral minority groups. Okay, so the next one is the newborns program. So at the moment, you'd all be aware that newborns have a heel prick test done, the so-called Guthrie test, which then has biochemical assays performed to screen for varying numbers of heritable diseases and newborn diseases like hypothyroidism, which are not heritable, with different numbers being screened in each country, but actually in the UK, I think it's 9 diseases that are studied through the Guthrie test. So we are running a research program over the next 3 years where we will be sequencing 100,000 newborns, and we will be reporting back to families actionable findings. So things where we can be sure of the diagnosis from the genetics, in other words, that they're high penetrance, where there's another test to back up your diagnosis, where there's a treatment or an intervention which will benefit the child if the diagnosis is made. And our modelling from that suggests that around about 1 in 200 children will benefit due to that. However, these people are going to be consented because this is what we hope might ultimately become a genome for life. So this will mean that basically these people will have this genome and will potentially be able to go back to it to identify pharmacogenomic variants throughout the rest of their lives. And so potential applications will include polygenic risk scores. Doug? We'll be able to take people from this with rare diseases in childhood and be able to feed into a drug development program for rare diseases in the UK. We'll be able to do recall by genotype research, so these people are all going to consent to being asked to consent to basically allow us to recontact them so that we can get more clinical data or to obtain biological samples or potentially to be involved in clinical trials. And we'll be doing some pharmacogenomics. The most obvious one there is the amikacin-related genes, but I'm sure that in time that list will spread beyond that. All right, so there are clearly substantial challenges for us in terms of how we actually achieve that, consenting, how we actually go about confirming the relevant variants. The data storage costs are substantial for 5 years, but then how do you do that for lifetime? How do you build that into access through electronic medical records and electronic prescription linkage? And then if somebody turns up with the USB stick to their general practitioner and says, here's my whole genome, how are we actually going to get around that difficult conversation and actually both educate GPs to say, well, that's not much use to me, mate, and also provide tools so that it actually can be used in routine clinical practice. All right, and then the last one, which is I'll stop after that, I promise you, is how might the sequencing be rollable out in terms of reactive studies as opposed to proactive pharmacogenomics or preemptive pharmacogenomics. And I think this is where our Cancer 2.0 program of putting local sequences in peripheral hospitals will roll out. So the reason we're doing this is actually for cancer, where some cancer types, like for example acute myeloid leukemia, you need a genetic test done within 12 to 24 hours to guide therapy. And that therapy might range anywhere from vitamin analogues to high-dose chemotherapy, dependent on that 12 to 24-hour sequencing output. And therefore, it's really important to get this done rapidly. And one of the delays that's involved currently is the difficulty of getting samples from peripheral to central hubs and then being run on large-scale equipment, which requires multiplexing in central hubs. So that slows it down. So it takes us on average 2 to 3 weeks to get back a pharmacogenomic test, whereas we know that with Oxford Nanopore technology, we can get it back within 24 hours for part of it, and a whole genome back within 48 hours. And so that does raise the possibility that you could actually apply this sort of technology, which is increasingly available across the NHS, for not just for this application, but also for infectious disease sequencing, to do pharmacogenomic variation in high-risk situations, such as, for example, pre-transplant. All right, so I'll stop there. Happy to take any questions, but lastly, just to make sure I acknowledge the people involved in that, and particularly Yvonne Leong, who did most of the analysis of the whole genome sequencing pharmacogenomic data. Thank you.